Hello, everybody. Welcome to the final week of Chem 100. Hope you're all doing okay. Hope you had a good Hanukkah and are getting ready for the end of the year. So we are going to finish up with our biological molecules, talking about amino acids and proteins today. And then on Wednesday, we'll talk about nucleic acids. And that'll be it. So I will also be getting ready for the final next week. So to help you with that, if you've seen on Blackboard, since everybody's done with lab, we have one extra day. We didn't really need to use it for stuff, but if you want to come in on Wednesday for a final exam kind of question answer period, if you prefer to do that in person, you need to sign up for a time because we can't have the entire class there at the same time. So there's a sign up link on Blackboard. You can either come to the first half or the second half, whatever you'd prefer. If there's not that many people, you're welcome to stay the whole time, but we can only have 12 people in the classroom at a time. If you have a lab that you need to make up, you can also do that on Wednesday because you can have other people in the lab. So we drop one lab from the semester. So if you only missed one lab, and then it's not an issue. But if you had to miss more than one because of illness or whatever, and you want to make that up, let me know. There's a couple labs that we can't make up because we won't have the supplies ready, but most of the other ones we should be able to. But just please ask if you want to come in on Wednesday and do that. Good question. Is there a handout? There is now. There was not. I thought I couldn't remember if I had one for this or not, and I couldn't find it. But like just before we started, I found it. So I will post this on Blackboard. You don't have to complete it and turn it in. There's no assigned handouts to turn in, but I will post this so that you can take a look at it after the video today. And I'll just use it for some of the pictures. All the stuff that's on here is also in our online book. So if you want to follow along, if you pull up the link from Blackboard, we'll be going through the book right here as well. And I'll be kind of flipping back and forth, looking at some pictures here and some over on the handout. All right. Let's start then. All right, so we've talked about sugars, we talked about lipids, and now we're going to talk about proteins. There's a similar relationship that we've already seen between small molecules and large molecules in each of these categories. The small molecules we sometimes call monomers, and the large molecules, when a large molecule is made up of a repeating small molecule, we call that a polymer. And so when we saw with monosaccharides and polysaccharides is probably the more correct way of saying that, but monomers would be like the simple sugars and the polymers would be the carbohydrates, the starches and cellulose and things like that. Although they're all really carbohydrates, so I don't really like this wording. I didn't write this particular handout, so I would call the monomers the simple sugars or the monosaccharides, and the polymers would be the starches, cellulose, etc., the polysaccharides. With fatty acids, that doesn't really work either. But we had a similar idea. It's a similar idea where you have smaller molecules that then come together to make larger molecules. I wouldn't really call something like a triglyceride a polymer because you really need many like hundreds of repeating units to get that. But it does absolutely fit when we talk about proteins. So proteins are made up of these small molecules called amino acids, whose structures we're going to look at in a little bit. And when those link together, we have a protein. So the protein is the whole big thing that's made up of these amino acids, very small molecules, just a few atoms connecting together over and over and over again. And we'll see what those look like in a second. Okay. So one important thing is just to think about all the different things that proteins do in living things. And this is, I think, a little bit biased toward humans in this particular chart, but all living things, much of what a living thing does in a very broad sense is controlled by proteins. So they can fit broadly into these categories, and you can probably figure out what most of those functions are just from the names. But you have structural proteins that give something structure. So something like keratin in your hair or your nails could be a structural protein. You also have contractile, so things that move move back and forth. Transport proteins, proteins that move things around inside your body. So like a hemoglobin protein transports oxygen. Storage, you can have proteins storing calories or storing other protein. Enzymatic action of proteins is really important. We're going to talk about that a little bit. We talked about catalysts in a previous lesson. So there's a class of proteins called enzymes that act as catalysts for biological processes. So things like breaking down carbohydrates, breaking down sugars, breaking down other stuff that you eat, also building things up, producing all the various molecules and parts that your body needs. We talked in the lipid chapter about phospholipid bilayers and how you need transport proteins to get through that. So that's up in the transport protein, but you also need enzyme proteins to actually put all that stuff together and do those chemical reactions. And then you have your protection proteins, like your immunoglobulins that help with the immune response. If you want some more details and examples on those, definitely take a look at the book, the online book. You can see that here. There's a nice chart that kind of goes through all that stuff and what those do and what some examples are of those different proteins. So like digestive enzymes, for example, amylase, they often end in ACE, A-S-E, which gives you a clue as to what they do. So amylases break down amylose, which is a starch. Lipases break down lipids. Pepsin is a protease enzyme, something that breaks down proteins, and so on. All right. Sorry, I keep clicking the wrong thing. Okay. So one somewhat interesting thing about protein in these days, and this is a little chart about how much protein you should consume per day. It's become a little bit of a diet fad right now to just consume tons of protein. You go down the supermarket aisles and all kinds of food are advertising how much protein they have. And with the idea that the more protein you eat, the healthier you are, or that protein is better for you than other types of macronutrients. That's not quite true. Proteins are broken down just by these enzymes and such, just like lipids and just like carbohydrates. If your body doesn't use them for building things, then it tends to break them down and use them for calories and storing energy, just like all the other macronutrients. The only people that really need a lot of protein are people involved in really intense exercise, muscle building activities that need lots and lots of supplemental protein. But most of us, I don't know about you, maybe you fit that category. I'm not building a whole lot of muscle here, so I don't really need that much protein. So just watch that. People are trying to sell you more protein than you probably actually need. All right, let's talk about amino acids, because that's kind of the basis for proteins here. So here is an amino acid. It's a relatively small molecule. There's a carbon. This A here is just a designation that this is called the alpha carbon. I think that's actually a typo. I think when I converted this over a PDF, it lost the Greek letter, but it's known as the alpha carbon of the amino acid. And there's another carbon atom here that is part of a carboxylic acid functional group. All right. Make sure you know those functional groups for the final exam from back in the organic chemistry chapter. You have a carboxylic acid, and then you have over on this side an amine. All right, so that's where amino acid comes in, the name. It's an amine and it's an acid. And then you have this R group. And we mentioned back when we were talking about functional groups, we used R as kind of like a wild card, like could be anything. And that's true for amino acids as well, but it's not that it could be anything. There are various things that it is, and that's what differentiates the amino acids from each other. So to show you them, I have them on this sheet here, and they're in the book as well if you want to flip through to that chapter. This is a little blurry. Let me see if it looks any better in the other. Oh, I still have the other thing on here. Hold on. Sorry. There we go. The problem with finding the handout right before doing this. Yeah, this one's a little bit easier to see. I'm going to flip it back again. Okay, so here's the chart from the book of amino acids. And so you can see here, here's our alpha carbon. In these cases, in these examples, we've got an alpha carbon here. We've got COO, which we know is actually C double bond O and then another O. And the reason that this is listed as COO minus, and then the NH, the amine part is NH3 plus instead of NH2, is that in a neutral solution like blood, because this is an acid with a relatively low, we haven't talked about this term pKa, but that acid, that hydrogen can leave easily. And then a nitrogen is somewhat basic. So actually in a neutral solution, that hydrogen's going to be gone, and this one's going to have an extra one. So the more correct way to express amino acids in neutral solution is like this. But we don't have to worry about that too much because every amino acid is the same for that part. The thing that's different is this R group, which here is highlighted in blue. So depending on what that R group is, gives us our different amino acids. So some of these names you might be familiar with. The simplest amino acid is glycine that just has an H there. Then you've got alanine if that goes up to a CH3. Valine if you've got 3 carbons, an isopropyl group there. And you can see like some of them get quite big with these big aromatic groups, which are these rings with alternating double bonds here. And so they are separated into these categories depending on the chemical nature of those R groups. And you should be able to somewhat understand those categories based on the stuff we've talked about this semester. So for example, here are non-polar R groups. Aliphatic, we didn't really talk about that term. That just means single bonded carbon, like we call them alkanes. But these are non-polar groups. So these are things with, just carbon and hydrogen. This one has sulfur there. Sulfur is not particularly electronegative, so there's not really much polarity there. Then you've got the polar R groups, so things with OHs, SHs, carbon double bond oxygen, so a couple amides here. Those are going to be polar. And remember this because it's going to be important when we talk about the larger protein structures in a little bit. Then, if I can move this over a little bit so you can see it better. Yeah, that's a little better. Okay. So then we've got the R groups that actually carry a full positive charge. So NH3 plus here, there's a plus, plus. So these are going to be extremely polar because they're actually fully ions. There's negatively charged ones again, or as well, aspartate and glutamate. And then you have these big non-polar aromatic R groups. Aromatic, again, is this term for these rings with alternating double bonds. So these are going to be generally non-polar, even though these two have OH and NH. If I don't, you can see that, I'll move out of the way here. OH and NH, they're still considered non-polar because there's so much more carbon and hydrogen compared to just that one OH and NH. So it kind of like overrides any polarity there. And those are the amino acids. So you don't have to memorize the amino acids. You should be able to identify the R groups based on their category, but you don't need to memorize anything for that as long as you understand the basics of polar and nonpolar. So if you have a R group that is all carbon and hydrogen like these, you should recognize that those are nonpolar R groups. Whereas if you have OHs, NHs, charges, things like that, those would be polar R groups. All right. Another word that we use, I think we talked about this last time, but just to say it again, in biological systems, we say nonpolar and polar, but a more common term to use is hydrophobic and hydrophilic. And we talked about this with lipids, I believe. Hydrophobic meaning nonpolar, hydrophilic meaning polar. Because living things are generally water-based systems, it makes sense to talk about that in relation to how it interacts with water. So things that interact well with water are hydrophilic, and things that are not are hydrophobic, or nonpolar would be hydrophobic. All right, so then let's talk about how amino acids come together to form proteins. So this is the condensation reaction that we've seen many times now in different situations. We have the acid part of the amino acid. This time it's written as the neutral carboxylic acid functional group instead of the COO carboxylate, just to kind of show this more clearly. But you take the OH and the H, get rid of those, and then connect what's left. And in this case, it's actually happening twice. So this one's happening in the same way. Take out the H and the OH, condensation or dehydration reaction, connect what's left. And you can do this over and over again because amino acids are functionally two-sided. They have an amine side and they have a carboxylate side. So if one of those sides gets used up in a reaction with another amino acid, that other amino acid is still going to have that other functional group on the end. So you'll notice here's a tripeptide, 3 amino acids linked together, but there's still an amine on one side and a carboxylic acid on the other side. So those can go on and form bigger and bigger and bigger in polymers. They can keep reacting over and over and over again. So that's how a peptide chain and ultimately a protein is built. This thing that is formed right here, I'm going to use a different color here so it is a little more obvious. So this bond right there is known as a peptide bond, which we also called an amide functional group. So amide is the more general term for any kind of carbon double bond oxygen with a nitrogen next to it. Peptide is specific to proteins. When we're talking about proteins, we call it a peptide bond. So we can continue to grow that. And then depending on what these R groups are, that's going to have a different meaning for what that chain ends up looking like. And so this first structure of the protein is known as the primary structure. We're going to talk about 4 levels of protein structure. The primary structure is which amino acids are connected together. So, you've got the amino acid, then that's connected to the next one, and that's connected to the next one. And you can actually list this whole big string of letters. Each amino acid has a 1-letter abbreviation. And often what happens when there's a new protein that needs to be understood is they will use some chemical and analytical techniques to figure out what is that exact order of amino acids. And that's known as the protein sequence. So sequencing a protein means figuring out which order the amino acids are connected in. And you're often talking about hundreds or thousands of amino acids connected together. So it's a long sequence. That's called the primary protein structure, and that is held together by those covalent bonds that we called peptide bonds. So those amide bonds forming from those condensation reactions. All right, so since this thing is so big, rather than just think about the individual atoms and how they might look, we can actually look at, kind of zoom out and look at the structure from further and further away. So the next level is known as the secondary structure. And I think this picture is going to get a little small, so I'm going to flip back over to the to the book. Ah, sorry. All right, so there's the chapter on primary structure, and we'll jump into secondary structure. So what happens is you have this big string of amino acids, and depending on the R groups, those chains can fold on themselves and interact with each other in some various kind of predictable ways. So, for example, if I can get this picture nice and big so you can see it, the protein or the amino acid strand can spin around and form a helix, and that's known as an alpha helix. And that forms because the protein structure or the amino acid sequence structure is arranged in such a way that these amide hydrogens line up with the carbonyl oxygens on a different amino acid and hydrogen bond with each other. And so we can actually make models of this. And there are some, one thing you can look up if you want to see this yourself, there are paper models. So you can print out a protein sequence and then actually cut it out and twist it and make it line up as an alpha helix. It's pretty neat. And it does line right up just like that. And that alpha helix is a really common type of secondary structure in proteins because amino acids can easily do this. The other one that we commonly see is called a beta sheet or a beta pleated sheet. This time it's from two side-by-side pieces of a amino acid chain. And I should say also, generally this is all one chain. So all of these things are part of one amino acid chain, but it's so long that it has all these different parts that can flip back on each other and all of that. And so that's the secondary protein structure. You can have this type of hydrogen bonding interaction, or you can have, which is called the beta pleated sheet, or you can have this type, which is called an alpha helix. Those are the two common types of secondary structure. And so when we zoom out a little bit from that protein structure, we stop showing the atoms at all, and we just show these shapes that you see here. So you see the swirl of the alpha helix, and these sheets are shown as flat, like arrow ribbons, because they are directional. They do have different sides, and that's why there's an arrow in them. And so when we zoom out a little bit, we see this secondary structure, which is caused or formed by hydrogen bonding. So the primary structure was formed by covalent bonding with peptide connections. Secondary structure is hydrogen bonding, which is weaker, but still a pretty strong interaction. We know hydrogen bonds, especially lots of them like this, can make a pretty strong structure. So that's secondary structure. The next level is other types of intermolecular forces that actually allow these beta sheets, alpha helix, other parts of the structure to connect to each other. And this is sometimes known as protein folding. So here's an example. Now you can see we're zoomed out even more. There's even less detail in our structure, but this noodly looking thing is the polypeptide backbone. So that's the helix, the alpha helices, the beta sheets, the other kind of swirly parts, whatever. And then emphasized here are the side chains, which are the R groups, that are interacting in some predictable ways. So if one amino acid has a positively charged R group and another one has a negatively charged, then those can kind of line up and attract each other. And that's actually going to cause that whole protein to fold in that particular way to connect those things. Similarly, you can have something called hydrophobic interaction. So if you have a nonpolar side chain like this one for valine, you can have two of those that might attract each other because we know that nonpolar stuff attracts other nonpolar stuff through dispersion forces. A couple other ones, there can be regular old hydrogen bonding. So if you have a side chain that has an OH, it can interact with another side chain that has an oxygen or a nitrogen and do that same kind of thing. And then another interesting one that's kind of very different from what we've seen so far is a disulfide linkage. So if you have two R groups that contain sulfurs, those can actually connect together in a SS bond between the two sulfurs. And this bond is not all that strong. So it's relatively strong, but it's kind of on the order of these other things. So not as strong as like a full covalent bond. It's a little bit labile. It can be connected and disconnected. And so all of these things together influence how that chain is going to fold up into a protein. And it's very complicated, and there's a lot of work that's being done to try to predict how these proteins fold and how they will do this, because that influences their function so greatly. You can totally destroy a protein by messing with this. That's known as denaturation. So if you add acid or heat, you can actually break these interactions and you completely destroy the protein. It no longer does what it's supposed to do. If you're interested in kind of a fun thing to do, one thing you can check out over break is this thing called Fold It. Fold It is a protein folding puzzle, and it's a game. It's basically a game. You get a protein and you're like playing with it and trying to figure out how to maximize the interactions. But the trick is you're actually doing real work also, because the best solutions to these puzzles are probably the actual ways that the proteins fold. So by trying these puzzles, you're actually helping scientists figure out what the best way for these proteins to fold is. And like at the beginning of COVID, they're trying to understand the proteins involved in the SARS-CoV-2 virus, and this was a big part of that people tried these various protein folding games, and they were able to figure out some stuff about how those proteins work. So if there are particular issues that are important to you, this is a way that you can help with those medical things, understanding different types of viruses, different types of proteins. And it's also just fun. Like you can give it to, you can play it with a kid, you can try it together. It's very much like just a puzzle game, but you're also contributing to figuring out how proteins fold. All right, so that's the tertiary structure. And then zooming out even more, we have what's called the quaternary structure. So if this is our tertiary structure, the thing's all folded up. Sometimes proteins consist of more than one amino acid chain, and those separate chains also interact and pack together. And so that's the furthest out quaternary level. We're not going to say much more about that. If we go back to the handout, which I will post this for you, you can see some of this. So if you looked up a protein structure online, you'd see something like this here with all these ribbons and swoopy things and different colors. And the way to sort of tease that out now is we understand that those ribbon curls are alpha helices. So that's the alpha helix. That's part of the secondary structure. The way that they're folded together is the tertiary structure. And then the blue and red are two different chains that come together to make this protein. So proteins are big, complicated molecules, but they're all made from those same 20 amino acids, just in different orders. All right. So the last couple things we need to talk about. I mentioned this just a minute ago briefly. Denaturation is where you disrupt those interactions. You're not breaking the protein chain. You're just getting rid of those secondary, tertiary, and quaternary structure interactions. And we actually used to do a lab. We didn't have time this year to do it, but you can do it at home. You can take some milk and you can mix it with some acid, and you'll see it like get all gross and separated. And that's because you're denaturing those proteins in the milk and then causing them to reclump up in a new way. And often when we see those sorts of effects, that's an effect of denaturation. You can also do that with heat. If you heat up milk, if you've ever made yogurt, you have to heat up the milk first. Partially that's for getting rid of bacteria, but more it's actually to denature those protein chains, those casein chains, so that they can retangle in a predictable way and make yogurt and make that like gel matrix that we all like from yogurt. Alcohol is another way you can do it. You can take, milk or egg or something, and you can mix it with high proof alcohol, and you can see some of that denaturation as well. And there's some other kinds of chemical ways of just breaking that apart. But it's mostly important that you understand that word denaturing, and just recognize that different chemical processes, when they interfere with hydrogen bonding, with nonpolar or hydrophobic interactions, with disulfide bonds, anything that interferes with those attractions is going to cause the protein to denature. And then we talked about catalysts before. So an enzyme goes with all of that. It just is a biological catalyst. It's a protein generally. And so, if you have a particular process that has a high activation energy, the catalyst makes it have a lower activation energy by going through a different mechanism. So enzymes are often drawn like something like this, like Pac-Man looking kind of things. And then we often say like they fit some other substrate that might look like, let's say we have some molecule that looks like that, and then you have this catalyst, and then it needs to interact with something that looks like that. So that can go in here, this can go in here, and the catalyst can kind of mediate that function, make those two things react with each other. Now, chemically, that's, what's really happening is functional groups. This is all involving functional groups connecting together. But many biological processes are would, in a normal, like if you were just going to mix these reagents in a lab, they would have a really high activation energy, and you can't have that much energy in a living system because stuff would be too hot. So you have catalysts, you have enzymes that bring that activation energy down and allow those reactions to happen at more normal temperatures. And so that's a very, very important function of proteins. And yeah, I guess that's all I got to say about that. So let me know if you have any questions about those things. Again, as usual, check Blackboard. Make sure you understand the main bullet points, the objectives that you need to know about proteins, so that you're ready for the final. Particularly, think about the functions of proteins, structures of amino acids, the types of side chains, because that, of course, all goes in with stuff we've talked about before, with polarity, with organic molecules, with functional groups, all of that. So, and then the 4 types or levels of protein structure: primary, secondary, tertiary, and quaternary. And I kind of went through it quickly here. You can look at the book for some more examples. You can also see some great videos on, if you look up secondary, tertiary protein structure on YouTube, you'll see some pretty neat animations of how these things actually fold, some of which are actual real, I don't want to say real videos of molecules, but they're based on real molecular data that is actually how those things fold. So check those out if you're curious about that as well. On Wednesday morning, we will look at nucleic acids. And if you want to come Wednesday for review, please make sure to sign up for a time. I know I've seen a couple people sign up so far. So that's great. And I'll see you then. Oh, and let me know if you need to make up a lab, and we'll see if that's possible as well. All right. Well, let me know if you have questions, and I'll see you Wednesday morning. Bye.